Hello, everyone. Thank you for joining us on World MS Day at the second MS Research Reception. My name is Darryl Dominic, and I'm the Director of Client Services and Government Relations for the Multiple Sclerosis Society Canada Manitoba Division, and I'll be your host for this evening. It's my pleasure to bring greetings this evening on behalf of the MS Society. I hope everyone's enjoying the tasty hors d'oeuvres and wine that's being served. I think it's a great service opportunity from the Viscount Gord Hotel. Let's give them a big round of applause. The MS Research Reception is the only reception on MS research in Manitoba. We are very excited to have with us today renowned speakers and experts who will provide the latest information about research in Manitoba and across Canada. Before we get started, I'd like to provide you with some details of the reception this evening. Washrooms are located directly across the conference doors. The presentations will be recorded for information purposes. DVDs of today's conference will soon be available to borrow from our MS library. I would like to take this opportunity to call to the podium Mr. Keith McConnell, Chair of the Manitoba Division Board of Directors.
I'd just like to take the opportunity to thank each and every one of you for coming out tonight. It's a beautiful night. I know it's the last night of one of the playoff series, so we're going to stay efficient and also provide an opportunity for individuals to receive this information via the DVDs that were mentioned. I'd also like to make special note of the number of board members who are with us tonight, some of whom are wrapping up their terms as board members and have dedicated the last 6 years of a lot of hours and effort to support events for MS, and I'd just like to take a moment to give those folks a round of applause and say thank you very much for everything. I'm always amazed when I come to these events. I look and I see so many familiar faces, and I apologize I don't remember every one of you that had a picture taken with me. I know that many of you have for raising a substantial amount of funds throughout the years, and your participation in helping us end MS and making the lives of those living with MS is greatly appreciated. So thank you to each and every one of you who participated in a variety of ways across the province. My heartfelt thanks to all of you, because it really makes this such a gratifying position. So thank you all so much. I just have one more housekeeping detail, because a number of folks approach me and say, are we having an annual general meeting anymore for the Manitoba Division? The answer is no. We've turned it into an information and research session like this evening, because as we've transitioned into a national board structure, we no longer have the fiduciary requirement to have a local annual meeting. That doesn't mean that we're not working very hard for you as a board, and working very hard to ensure that the services that are provided to Manitobans living with MS continues, that we continue to raise money in Manitoba, and that we do research in Manitoba. So part of our focus is to encourage that, but I also wanted to make sure that I cover that off, because I know a number of people come and approach us on a regular basis and is there a general meeting? This is our general meeting. We're transitioning to awareness and getting the message out that it'll take each and every Manitoban to help us end MS. So thank you all very much for coming tonight. I know that you will enjoy these amazing speakers, and I'd like to thank them also. We've had some speakers who have come from far and traveled hard, and we appreciate that. As well, I'd like to thank the renowned speakers for, and I don't want to disrupt Darryl's introduction of their CVs, so I'll hand it back to Darryl. Thank you all very much.
Thank you, Keith. Connect. Connect is a single word that can apply to many different situations. It's all here today, it's all about meeting others who have an interest in ending MS. To me, Connect is about joining with others who are realizing what is possible with MS. From the thousands of volunteers who work tirelessly on our annual MS Walk event to sponsors who contribute generously so that our conference runs successfully. From the dedicated researchers who are making great strides towards better understanding of why MS develops, how it progresses, and how neurological disabilities might be prevented. To the healthcare professionals that help with disease and symptom management, enhancement of function, and improvement in the quality of life of people living with MS. People living with MS are reclaiming a sense of dignity, control, and freedom, and empowering themselves with the knowledge, skills, tools, and confidence to transform challenges into possibilities. In the last decade, a great deal has been accomplished by our dedicated researchers in Manitoba and across Canada, and these advances continue to offer hope for everyone affected by MS. Tonight, at this moment, we are all connected. It's so important for you to know that 50% of monies raised go to programs and services for Manitobans living with MS. The other 50% goes to support groundbreaking MS research in Canada, some of which is actually happening in Manitoba. I'd now like to introduce our first speaker for the evening. Dr. Jorg Stetefeld is the Canadian Research Chair in Structural Biology for the Department of Chemistry and Department of Microbiology at the University of Manitoba. Dr. Stetefeld studies the structure, function, and regulation of proteins involved in the extracellular matrix that surrounds cells in humans and other mammals. His primary goal is to understand structure-function relationships and protein dynamics, and thereby provide fundamental knowledge that could, for example, be used to develop practical treatments for a variety of different diseases. Notably, he has successfully crystallized netrin, a protein that guides the growth of axons, a fundamental part of the human nervous system, to allow for its study. The expression of netrin is crystallized form in quantities sufficient for analysis and has been a long-standing problem. Understanding how netrin works has the potential to better understand and treat certain neurological disorders, much like MS. Please welcome Mr. Jorg Stetefeld.
Don't worry, it sounds more complicated than it is. We see, I live in a very, very simplified. I'm living in a very simplified world.
Technical difficulties are ours, folks.
First of all, I'm very pleased to be here with you tonight. The years before I moved to Canada with the family, I worked for 10 years for the pharma industry in Switzerland, partly for a company called Hoffmann Laroche, and then for other spin-offs and at the university. And there, I have to say, it's a complete different culture. Our research is funded, and our researchers like me are engaged to be in contact with people we are technically working for. And most of the time I was in charge of a program to cure early childhood muscular dystrophy, a disease which in special areas in the Alps, Switzerland has the Alps mountains, is causing terrible high death rates. And my job there was to crystallize proteins and to help to develop drugs, which finally led to the foundation of a company called Contract, and then to the first cure of a Duchenne kind muscular dystrophy. And there we were always engaged to meet kids in the children's hospital. And I have to say that this is something I miss a bit here in Canada. It's the first time after 7 years that you guys asked me to come and talk about what we are doing. What I appreciated there was always this kind of a special kind of spirit, and this kind of meetings can create a certain kind of motivation for the work. Okay, what we are doing. You might have heard, you all are aware of what means muscular dystrophy, but most of you might never have heard the name structure-based drug design. And I assure you, I have 4 boys, and I had to give this talk in front of them, and my wife was very, very harsh, and I tried to do it as simple as possible. But if you feel, what is this guy talking about? You are more than minded. There is sometimes the reputation of our institution can be improved, but that's great research going on. Also at the University of Manitoba, and there is science rendezvous, and there are open days, and if you ask me in advance, we can also arrange a lab tour, because I came here 6 years or 7 years ago, and Canada invested a lot of money in my research, and there's a lot of things going on. And this is just a success story which has nothing to do with the multiple sclerosis, but it shows you how I think and how I work. This is a platelet. A platelet is a thrombocyte which technically is there to form clots. And these clots are required to stop bleeding. And you and me undergo a permanent turnover in decisions between aggregated and non-aggregated. And at the surface, this platelet has a receptor, which is called integrin. This is what happens if we expose this kind of integrin receptor with its target, which is a collagen. I don't go into the intimities why collagen, but you see here, if the substrate bound to this kind of a receptor, the thrombocyte is aggregating. This is the starting point for the clot. What we do, and this is the scheme of this kind of a receptor, it's an integrin, which is thousands or billions of times at the surface of this kind of a platelet. What we were interested in to control this mechanism. And we were contacted in 2007 by the director of the state snake venom laboratory in Sao Paulo, and they approached us with the snake. It's a so-called pit viper. They saw this pit viper in its snake venom is secreting a compound which is extremely poisonous to its victim. An adult man or woman would die within less than 5 minutes, because this snake developed an extremely smart strategy to exactly fool around with this kind of mechanism. What we did here in Winnipeg, in collaboration with people in the physics department, they are the big guys in developing certain technologies like handstanding. We analyzed, we got the raw snake venom, which was a lot of paperwork to do to get this material into Canada. We isolated a compound which we later on named rhodocetine, and we crystallized this compound and analyzed it. And to make a long story short, this is a clinical trial drug now for the company Aventis in Germany and France. What we saw is that this rhodocetine, this red and yellow stuff here, is what the snake injects in the venom or in the bloodstream of its victim. This kind of lectin is blocking excess of the natural substrate, which is collagen. Technically, the victim is bleeding to death. However, this is a known or somehow familiar mechanism, but what was unknown, that this was the target or the starting point to develop a drug, was that this snake venom, under certain circumstances, they are washed off, after a certain time frame. However, this guy is so smart, he changes this little yellow ball here. Usually you and me, we have their bivalent cations like cobalt or manganese, and the snake is changing them to calcium. It means all the integrins are never ever able to interact with collagens anymore, and technically the victim can never run a blood coagulation cascade. And this is, so to say, the world I'm in. If I do science, we dissect things to a very, very molecular level and try to understand how it works. And sometimes I'm asked, if we go to companies or we deal with vendors or so, what is the approach? And perhaps the only thing you might remember today is I'm a guy who works in the structure-function world. We try to determine the structure as precise as possible, and with this I really mean we go into atomic details. We really, I can tell you at the end of the day, there are carbon, oxygen, you name it, atoms. From this we understand the function. We try to learn about the function, and this is for us always the starting point to develop applications. There are different ways to describe the disease, multiple sclerosis, and as you might realize, or have realized already, I'm not a medical doctor, I'm a biochemist and crystallographer, and we try to really nail our work down to the atomic, to the molecular problems in a disease. And we are all aware of that technically MS is a demyelination problem. at a certain point, patients can't repair the myelin sheath surrounding the axons anymore. There are different arguments about the molecular mechanisms, what's causing this, and this of course would be for someone like in my field always the first point of attack to understand what's the reason. So far, drugs available on the market developed mostly or targeted in the third row of attack, namely that they try to block inflammation or inflammatory responses. But we are a group who is technically interested in, if this myelin sheath, the surrounding material around an axon is damaged, how can we repair it? And Mother Nature already has a system in place, and these are the famous oligodendrocytes, which are cells surrounding axons, and their job is to produce the myelin and to secrete it and to allow the formation of the myelin sheath, the surrounding element. I know we don't have the time to go into all the details, and especially the maturation of these oligodendrocytes is something of extreme importance these days. Technically, I'm not working with oligodendrocytes. We are interested in their precursor cells. But as you have seen, this integrin, the receptor on a platelet, also these guys have at their surface, anchored in a cell membrane, they have receptors. And receptors always means these are proteins talking to the surrounding environment, and they are extremely important to us. And we have two things here to consider. These oligodendrocytes, or the OPC, sometimes they have to move to an axon, which is called the so-called chemoattractive response. And this is driven by a receptor called neogenin. On the other hand, sometimes they don't. It's dangerous if they are close by. They have to be repelled. In this process, it's called chemorepulsion. And this is driven by another receptor, which is called UNC5. The names doesn't matter at this point. But the question, of course, who is telling an OPC or an oligodendrocyte, go there or don't go? And the guy in town, or the system we are interested in, who is really the chemoresponsive axon guidance molecule? And Daryl named or mentioned the name already. The molecule is natrin. And so far, there was no know-how about how natrin looks like. We come back to the structure, function, application theory, and this was, so to say, the starting point for me to step in. Okay, now I'm a bit too small. This is, my field. I'm a crystallographer. This means we produce proteins and then we grow crystals out of it. And I know it's not fair. If it would be darker, you would see this is a protein crystal of natrin grown in a time frame of about 5 years, which is, of course, something very, very demotivating for a graduate student, but at least we were lucky and we are still the first one who could crystallize the system. After a lot of work, this is the second version of a natrin crystal grown shortly before I moved to Winnipeg from Basel. And this is a guy who grows in about 2 years. Now we have optimized the method in a way that we can grow these little crystals, which are not visible with the naked eye. You need a reasonably good microscope, and we can grow them in about 6 months. A crystal trail here, usually we make them in the upper 10,000 of experiments, are extremely expensive and need a lot of experience and hard work. What we do then, usually we go to Saskatoon, but because I had the access to, or I had the opportunity and the honor to receive the Canada Foundation of Innovation grant, we have an in-house source here now in Winnipeg. So to say, we take these crystals and expose them to extreme high energy X-ray beams. And this is the method I am using to get these pictures. You might say, come on, what's all about this pink, blue, and yellow stuff? This is an atomic picture. You can't be more precise than this of natrin as a molecule. I can't go into the details. What we did subsequently is we took this molecule and nailed down its diverse binding activities to both chemoattractive and chemorepulsive receptors. And that's why I highlight this here in pink. This is exactly the region where natrin, if it binds to neogenine, causes repairing of myelin sheath. And this is if natrin binds to UNC5, it's a repulsive element which causes certain problems in the OPC maturation. And last but not least, what we then do, we take these proteins, we take natrin and we take the receptor and study their complex. And this, whenever you will see these kind of pictures, this is a starting point to develop drugs in a so-called structure-based drug design approach, because at this point you have a molecular picture at atomic level how natrin talks to one of its receptors. And as a starting point, it's a bit too early to go into all the details. We designed so-called peptidomimetics, where we mimic, so to say, the miniature version of natrin and perform so-called axon guidance assays. And just by producing, by knocking down, or with the silence interference RNA technology, we diminish the functionality of natrin, and then we add to a certain assay the drug we are interested in, and then we can see a little peptide here composed of a couple of amino acid residues. When we add certain lipids and certain sugar moieties, allow to reinforce the structure or the function of natrin. And this is for us, of course, an extreme strong starting point to develop more enhanced mechanisms or small molecules which might lead in drug development. Another thing in respect to all this crystallography is that, as I mentioned to you, it's in the moment the only technique available on planet which allows you to produce atomic detailed information. You might have heard 2014 is the year of crystallography. I mean, it's honored with more than 52 Nobel Prizes. The last 2 in chemistry were given to people working in this kind of field. They work with different systems, but it's still an extremely powerful technique to study really the atomic detail, which is a molecular fingerprint to develop a drug. And what we see here, this is an electrostatic potential, just very, very brief. You see here a lot of blue guys and some red guys. And this is for us a tool to develop drugs, which specifically blue for me means it's positively charged. It means we design drugs based on heparins and heparan sulfates, which enhance the activity of this small molecule. Last but not least, and this is something where even the guys at Montreal or Toronto are coming to us, we got funded from the NIH and other organizations to get a machine here. And there we are the only ones in Canada who can do this kind of, it's called a small angle X-ray scattering approach, where we can study molecules at semi-atomic resolution in solution. We don't need a crystal. And this what now was the big surprise, and that was the reason why I have to go tonight to Montreal, because there we use this kind of know-how now to test certain compounds. We realized that we have an atomic picture of natrin now, but this is not the complete picture in nature. And the picture in nature is that natrin always exists as a dimer. There's never a single monomer of this molecule. And this dimer formation is of extreme importance to its function. And as you see here, these dimer molecules of natrin are arranged in a head-to-tail antiparallel manner. It means if this is one natrin, the other natrin is arranged in this way. They are not like this. They are antiparallel arranged. And this is we now develop mechanisms or tools to destroy the dimer. Because we have an interest to allow the proper maturation of OPCs, or we develop tools to enhance the dimer formation. But interestingly enough, the fact is that even in this dimeric stage, in its natural conformation, natrin is fully functioning. And you see here, this is a model we generated with our in-house technology where one netrin dimer, this blue guy, can bring two neogenin, two receptors, two dependence receptors responsible for the chemoattraction for moving oligodendrocytes to the target of myelin sheath damage and allowing there to start the repair. I know lots of facts, and I hope it wasn't too complicated. I didn't waste you with all the technical stuff, but you can imagine. You haven't attended a lecture of mine. That's why my second evaluations might be improved a bit. Okay, this is a team. I really had the great honor to attract a couple of guys from Basel joining me. I'm very happy that this gentleman, Marcus Meyer and George Orris, he's an Englishman, are still with me. We work already together in the industry in Basel. And there is always a bit, I have to say, we never had problems to attract students from within Winnipeg, and I always had the philosophy, come on, I'm now here in town, let's look for the kids in the city. There are lots of negative things about us, but I have to say, Matt McDowell, Natalie Oku, now Pran, Oksana Marusha, Kelly Edwards, and Brigitte Bauer are Winnipeggers, and they are very, very hardworking, and it's a great team to be. And Tusha Patel is now a Marie Curie Fellow down in Birmingham, UK. You can imagine this is something, if you try to develop a drug, how we do this, we are only a part of a team. We are a part of things. We technically are the guys who make the pictures. We make very precise pictures, but we can give suggestions, and we can design, and we can synthesize small molecules as we did already. But I'm not a medical doctor. I don't have, we need partners to do this. And a very, very great gentleman, one of the guys we found, naturally, Peter Yurchenko from Piscataway, New Jersey, is doing all sorts of in vivo studies with us. And a fellow I work with in industry, now he's in the Biozentrum in Agri in this muscular dystrophy project. And a gentleman from Germany, Manuel Koch, we work together. And yeah, that's why I'm here. This research needs money, and a lot of money, and I'm very pleased and very thankful that the Multiple Sclerosis Society of Canada thought it's worth investing in me, and I'm also thankful to the Canada Research Share Program. And thanks for your attention.
Thank you, Dr. Stetefeld. We're going to reserve questions until the end of the presentations, so if you can keep questions in mind, we'll be eventually putting out a microphone to folks who want to participate in that process. I'd now like to introduce our second speaker for the evening, Dr. Donna Wall. Dr. Donna Wall is a professor of pediatrics and child health, immunology, and internal medicine, the director of the Manitoba Blood and Marrow Transplant Program, director of the Cellular Therapy Laboratory, and member of the Regenerative Medicine Program. Dr. Wall returned home to Manitoba after many years of practice and research in New York, Boston, St. Louis, and San Antonio to head up the Manitoba Blood and Marrow Transplant Program. Her research interest is broadly focused on the field of clinical blood and marrow transplantation, with a focus on the immunologic aspects of transplantation. Within the Manitoba Blood and Marrow Transplant Program, she and her team focus on addressing issues of care delivery for Manitobans. Dr. Wall's laboratory focuses broadly on developing cellular therapy in Manitoba, and specifically on exploring the possibility of using immune regulatory cell populations as treatment for inflammatory disorders such as multiple sclerosis. Please welcome Dr. Donna Wall.
Thank you for the kind introduction, and really a pleasure to be here. Multiple sclerosis is a problem that's near and dear to my heart on a personal level, and I tell you, I'm a little nervous. I'm a bone marrow transplant doctor. My zone is in blood making and the immune system, and so this is a little bit off my territory. What I'd like to do is take, since I have the podium and you can't get rid of me for a few minutes, is to take you through a little bit of a journey to explain stem cell therapy, what is involved with stem cell, and kind of to highlight what the direction that we're going, but also to highlight what is really involved and the complexity of what's involved, because right now you can probably just go to the newsstand and pick up a paper, a journal, a newsprint saying stem cell cure for you name your disease, and often it's multiple sclerosis. And I just want to give you a flavor of what's actually involved and the things that we're thinking about in the laboratory. The promise of stem cell therapy is huge. We're all aging, as much as we like to avoid that. And as we age, we lose the ability to regenerate, to grow, to recover function. It applies across all parts of the body, all organs, all tissues. Specifically for multiple sclerosis, when we look at stem cell therapy, we're looking at two things. One is that we want to stop that inflammatory process that's going on and the damage that's caused. And the second is, ultimately, we'd like to talk about repairing areas of damage. Okay. The journey to move towards a successful therapy is a complicated one. Okay. It involves first of all, finding a cell source to recover the area of damage. All parts of the body have stem cells with them. So you're going to find me getting probably almost even angry when I hear people talk about stem cell therapy. Don't you know what stem cell means? And I said, I really don't understand what stem cell means. There are cells that have the ability to recover function. They're very quickly in the body, specific to areas where they live. And I'll show you the example shortly. Once we isolate a cell that has the properties that we want to recover and repair, we need to grow it up in the laboratory, get it ready to do something good, okay, so optimize it. We then need to figure out, I have something in my test tube here or in my culture plate, and I have somebody who's sick over here. How am I going to get those guys to go from the culture plate and get to the place of action, into the nervous system? Okay? And so do I put it in the bloodstream? Do I put it in the spinal fluid? Do I inject it into the brain? If I do that, how do I get those cells to know how to go where they're needed, to set up shop, and to live long-term, so long-term engraftment. We have in the body an ongoing system that says, okay, this is you, this is me, you belong here, you don't belong here. And that process is a good one, because if we make mistakes in our body, we don't want those mistakes to stick around, because that's called cancer. But we want those cells that we put in the stem cell therapy to know how to get where they want to go and how to stay there and grow and function within that organ. So it's really a very complex process, taking cells that have stem cell function in them, and stem cell meaning they can reproduce and recover and regenerate function, and to get them to where you need them to do what you want to do. I'm a blood doctor, and what I do all the time is bone marrow transplantation. And so what I do is I take the hematopoietic stem cell, which lives inside the bone. So next time you have chicken and break open a bone, the mushy stuff is our bone marrow. That's our factory for making blood. And these young blood-making cells, if we transplant them from one person to another, and we're lucky in bone marrow, I can just put it in as an IV. They know how to get into the bones. Don't ask me how. It works. They then they'll grow and grow and grow and make all parts of the blood. You heard a little bit about platelets today, red blood cells, immune cells. Sitting inside that mushy bone marrow is a population of cells that their normal function is to support the blood-making stem cells, okay? And so these guys kind of sit up next to the blood-making cells, they nourish them, they give them signals to say, okay, you sit here, chill, we'll take care of you. They're kind of sheltering kind of cells. But when we need blood, when you need to make red blood cells because you're a little anemic, or you've had a nosebleed and you need extra platelets, these guys will say, okay guys, you go out and do your stuff. You start dividing. And these supporting cells are called mesenchymal stromal cells, or stem cells, okay? So they have the word stem, okay? You've heard me in this talk so far talk about hematopoietic stem cells, mesenchymal stem cells. These mesenchymal stem cells are a fascinating cell that have tremendous properties. If I put a needle in your hip or my hip and pull off a teaspoon of that bone marrow and put it into culture in the laboratory, these guys will slowly grow out over a period of a couple of weeks and look very bland looking, just long kind of stringy, chill kind of cells. They grow over time in culture. They'll still look like this. We know how to harvest them. We know how to collect them. And they have features that are absolutely fascinating. They will, if I give them the right signals, they'll make fat. None of us want extra fat in the body. They'll make cartilage. They'll make bone. They'll make skin cells. And in the laboratory, I can get them to have features that sure do look like neural or nervous system cells. And here's a picture of one of the cells that we had labeled up with a long, finger with the right kind of receptors at the end of it. They're not normal. They're kind of expressing multiple things at the same time. But these guys have that potential in their machinery, given the right signals, to become all these mature cell populations. But if I took those mesenchymal cells from my culture dish and just shot them into your eye, into your arm, into a vein, they're going to circulate through your blood-making system. They're not going to figure out how to get up inside the brain when they're needed. They're not going to have the time to set up shop and mature up and do the kind of things we have. Not with our current technology. We have research going on in the lab with collaborators looking at ways of getting these cells to become more purposeful, to learn how to get to where they're going. But what we are very interested in is specifically in the potential treatment of disorders of the immune system, such as MS, is that these mesenchymal cells, those very boring looking guys who look like they're not doing anything, are potent regulators of the immune system. They can shut off the strongest immune inflammatory response that you've ever seen. And they do this with multiple attack plans. They function on all arms of the immune system. And if you think of it, why would the body have that? Usually things make sense. So if you think of it, our bones are a small area, okay? They don't get larger. And we have that the inside space of bones is a really kind of compact space. And the job there is to grow immune cells. So these guys have, as they're talking and keeping the young blood-making stem cells chilled and relaxed, they have another job saying, I don't want a big inflammatory response inside the bone marrow. Bad things will happen. We won't be able to make blood. So this is kind of an accentuation of their normal process. We can grow them up in the lab. We're good at doing that. And some of the work we've been doing has been to characterize them and to learn how to get them really excitable and teased up in action. These cells have been shown to have such good immune fighting response that we're now looking, we being the international MS research community, at testing whether these cells have the ability to slam shut that inflammatory response that is driving MS. Okay? And this is a trial called the MESEMS trial. We're working to help get things set up and help some of the other researchers with this. And this is a trial that is involving patients across the world, centers in Europe and North America, looking at saying, if we take these cells and we inject them into patients, can we stop that inflammatory process that is driving MS? And so the way this study's done, because it's very hard, this is the hardest disease to say, is my treatment working or not? And am I getting better, or does the disease just decide to get quiet on its own, or did the disease kick up and I'm really having an effect, but it's not as a big effect as I would like? So this is a trial that involves a randomization. And so what happens is patients will be approached with certain type of MS. They'll agree to participate in the study. We'll, because we're blood doctors, this is what we know how to do, so we're just part of the team. We'll pull a sample of bone marrow, roughly 3 tablespoons worth, take it to the laboratory, grow it up, and freeze it. And then the patients will either receive cells right at that time point, or they'll wait 6 months and receive cells 6 months later, but in a way that we have a placebo. So, you know, the worst people who can't keep a secret and are so biased wanting things to work are the doctors and nurses. So we have to be blinded in knowing who's getting the treatment and who isn't. And so there's a randomization, and then 6 months later, the patients will receive the other treatment. The goal of this study is to use MRI scan, imaging of the brain, to see whether we decrease the number of inflammatory lesions in the brain. And so this is honestly an international trial. The leads on it are Canadian Dr. Friedman in Ottawa and Dr. Ukeli in Italy, and it's one of these really beautiful international collaborations, and we're really proud to be helping out. What's important with this is that even though we are giving mesenchymal stem cells, we have the magic word stem, and even though in the test tube, in the laboratory, I can make these cells look like neural cells, we're not expecting these cells to repair in the brain. Okay, we're not expecting these cells to grow new neurons. What we're expecting them to do is to work with other components of the immune system, quiet down the immune system, give factors to help in the healing process, and hopefully just shut off that, whatever that thing that got turned on. Is this study the end study? Is it going to be a cure study? No, it's the beginning of the end, to quote my friend Winston Churchill. We're anticipating that ultimately we'll need different approaches. We'll need multiple infusions, possibly. I can make some postulates on that. But this is very exciting for us. It's a first foray into cell therapy for treatment of MS. So in conclusion, the regenerative cell therapies are complicated, and they involve multiple barriers. Those cells are going to be expected to go through multiple different treatments, different steps in getting to where they're wanting to go. So it's very important as you read every day of new cures, cell cures, stem cell cures, that you feel free to call anytime, but that you take a look at what is really there. I've taken care of many patients who with my kind of patients and with my kind of disorders who have traveled far and wide for stem cell therapies, and often they've really not gotten what they think they've gotten. The MESEMS trial, as we call it, is a first step to introduction of cell therapies in multiple sclerosis, but this is going to be a journey. It's not a one-stop. So I too am just speaking for a team, and it's a wonderful team. We are working with the multiple sclerosis clinic. Our team is the Manitoba Center for Advanced Cell and Tissue Therapy. I've got Dr. Szwacer is a good colleague. I've got my trainees. We're looking at understanding these cells getting a really good understanding of them, and so working with the Manitoba Proteomics Center, Dr. Wilkins, and our new collaboration with Dr. Malcolm Jing in the engineering department, looking at smarter ways to grow these cells, smarter ways to protect them, coat them with nanoparticles so that they're going to be able to sneak in and get to places that otherwise they couldn't get to. We work closely with the team at the Ottawa Hospital Research Institute. I'd like to say thank you to the MS Society and Manitoba Health Research Council for support in helping us get our laboratory to the stage that we're going to be able to grow cells for clinical use. So I'll stop at that point, and thank you.
Thank you, Dr. Wall. We'd now like to introduce our final speaker, Dr. Karen Lee. Dr. Karen Lee is the Vice President, Research and Managing Director of the MS Research and Training Network for the National Office of the MS Society Canada. In her role as Vice President of Research, Karen is responsible for and working as part of an exceptional community of clinicians, scientists, and MS staff, providing leadership to the research programs of the MS Society of Canada. She also oversees the day-to-day operational leadership of the broad range of the Society's research program, building on Canada's leadership role in finding a cure for MS. As a key contributor to this effort, she supports senior volunteer committees of top scientists in the field in Canada and internationally. Karen obtained a PhD in cellular molecular medicine from the University of Ottawa, or a doctoral studentship from the MS Society of Canada. She also holds a Bachelor of Science honors in cell biology from the University of Western Ontario. Please welcome to the podium Dr. Karen Lee.
Hello everyone, it's a delight to be here again. I was here last year and I thoroughly enjoyed meeting all of you. And once again, I'm sorry if I don't remember your name, but many of you I do recognize. I won't speak too long because I think I want to give you the opportunity to ask quite a few questions to our researchers because we are running a little over time. But I do want to give you a bit of background in terms of where we're going at the MS Society in terms of research and what we're looking forward to in the next few years. One thing that is really dear to me is this. So this is something I always show because this is what got me interested in research, and more specifically in MS research. I'm not a crystallographer, but I was very interested in similar things as Dr. Stetefeld in terms of how cells talk to one another. For instance, his netron is on the cell, and it might want to talk to another cell, but how do they do that? So that's really why I got interested in MS research. And in fact, during graduate school, I always showed this picture, and I'd explain multiple sclerosis is a disease that affects the central nervous system, the myelin is affected, and therefore people living with MS might have mobility issues and then eventually paralysis. And that's how I would explain multiple sclerosis, a very textbook form of MS. And during my studies, you do spend a lot of time in the laboratory and also at a desk. It's a lonely time sometimes. But I was really fortunate to live in Ottawa along the Rideau Canal. Has anyone lived in Ottawa before? It's a beautiful city, right? Just like Manitoba here in Winnipeg. There's this beautiful canal, and one day I happened to be sitting at my desk that faces the river, or the canal, which is really fortunate for me, and there was an MS walk that was going on right in front of my place. And there I was doing research on MS, and I didn't know a single person living with multiple sclerosis. Yet here there was a group of people, just like you, fundraising for MS research for me, allowing me to do the research. And that's really where I realized there was a bit of a disconnect for me. I was in the laboratory working with mice all the time, trying to understand multiple sclerosis, but I didn't really understand what it was like to live with MS. So therefore, I started getting more involved with the MS Society of Canada at the Ottawa chapter. I had the opportunity to meet someone who actually went through a stem cell therapy. Her name here is Jennifer Molson, through a trial with Dr. Mark Friedman with a bone marrow transplant. At the same time, meeting someone who lives with progressive MS. And there's Marie Vaillant on the left, and she's someone who has lived with progressive MS for many years. And from there, I started volunteering, doing the bike tours, and really getting to understand what the organization was all about, but more importantly, meeting the people who were living with multiple sclerosis. And with that, that's really something that we're really trying to drive more of within the organization in terms of the research community and the connection to people like you. So just to give you a really quick overview, the MS Society actually has multiple programs of funding, so where do all the dollars go? This is just a really quick snapshot of what that looks like, in the sense of that we always fund researchers from a very young age, from undergraduate at a university to the more senior level, like Dr. Stetefeld and Dr. Donna, to do research within the laboratory. And by having this, what we call our pipeline, we're always ensuring that there's a good mix of MS researchers from young to more senior. I don't use the word old anymore. I got rid of the people upset, so I'll say senior. Is that a good word? No, maybe not even. But anyways, or more experienced, shall we say, seasoned researchers, to ensure that we had an excellent pipeline of MS researchers here in Canada. And in fact, annually, we fund approximately 6 million, and sometimes 10, depending on the certain projects that come through. But this really allows us to foster the great MS research that's ongoing here in Canada. At the same time, we offer what we call national education and training programs. This is a very interesting initiative. It's called the NMS Research and Training Network. Not only do we provide funding for researchers to do their research, so just like building or doing crystallography, that requires a lot of money, to providing studentship, so allowing the student to actually have a salary, but we also provide education programs for students to learn more about multiple sclerosis, whether it is in a different field that they're not familiar with. So for instance, many of us work with mouse models, but we don't necessarily do imaging in the brain. So how do you learn about that? We provide workshops on that. And so more recently, we had the NMS 2013 conference in Quebec. And you can see here, this is an amazing group of young researchers that came together from all across Canada, and every single one of them are doing MS research. And the type of research that they're doing, it varies from crystallography to protein to understanding how cells talk to one another, imaging to rehabilitation. So they spent 4 days all together. These are just the young trainees. We also have the experienced researchers who aren't in this picture. But that is your next generation of MS researchers here in Canada. And so that's an amazing bunch of people. But we offered them many other programs that allows them to learn more about what it is that multiple sclerosis is, so that they can bring that back into the laboratory and really make an impact in MS. More recently, what we're really trying to do is when researchers talk, you're probably very familiar that we have a really hard time talking about science in layman terms. We just assume everyone knows what DNA is and protein, right? But it's not normal to actually know what DNA and protein is all about. So what we're doing a lot of is workshops where young students get to meet people living with MS and tell them about their research. And that person living with MS will say, I have no clue what you're talking about. And then therefore they get feedback so that they can further explain what their research is about. And I think that has been a really big lesson for us at the organization, is we fund over $140 million of research since 1948. But we've had a really hard time explaining what exactly that is. It's not that we're not doing MS research here in Canada, we're doing a lot. But being able to explain that to someone who doesn't have a science background takes a lot of skills. And I have to say, the researchers are amazing in the laboratory, but being able to talk about it in layman terms is the most difficult thing on earth. And so we're really working hard with researchers to make sure that they also have the tools to understand how to explain it. And so with that, many of you may have participated in our research priorities discussion last year, where we went across the country, we brought young researchers and people living with MS to talk about what MS research means to them. And throughout the country, we heard loud and clear that MS research means hope. It really means hope. So in order to ensure that there is hope being generated, we don't just put dollars in, we want to make sure the communication is continuous, that we have the person living with MS involved in research, and at the same time the researcher understands what it is like to truly live with MS, to ensure that their research has impact on the people living with multiple sclerosis. And also, throughout that research discussion, we came up with top priorities. for the organization in terms of research. We heard loud and clear cause is a major issue. We still don't know what causes multiple sclerosis. There's many theories out there, and we want to further understand that. At the same time, progressive MS. We've made leaps and bounds in terms of relapsing remitting MS. Currently, we have 10 therapies, which is amazing, because 10 years ago, there were 0 therapies, but now we have 10. But in terms of people living with progressive MS, we have 0 therapies. And we understand that is a really important aspect we need to further explore. As well, we have diagnosis. We want to ensure that we have quicker diagnosis, so it no longer takes maybe the second attack. We could potentially have a blood test to diagnose multiple sclerosis, as well as repair. We've heard the importance of repair. Right now, all the current therapies suppress the immune system, but how do we ensure that we can bring myelin back, that we can have healthy myelin being reformed? We also have what we're calling life-modifying therapies. And this is a term that I heard out in Edmonton, and I'm going to continuously use that. And what does life-modifying therapies mean? It means for the person living with MS, I am willing to take those disease-modifying therapies, but what can I do in terms of my quality of life? Exercise, diet, meditation, yoga, which I know is very big here in Winnipeg, at the Winnipeg office. What can we do in terms of that, in terms of lifestyles, to ensure that we live a better quality of life? So that's a really important aspect, as well as cognition and mental health. We know cognition and mental health is a major issue in multiple sclerosis. How can we ensure that we're supporting people living with MS in terms of cognition and mental health? So those were the top priorities that came out. But what also came out for us is we heard we need to do the research now. There is an urgency. And we've heard that loud and clear. Now, as much as I would like to breathe down all the necks of the researchers, and I know all of you, to do the work faster, they're already doing the work extremely fast. I can tell you right now, there's probably a graduate student sitting in the laboratory looking at a microscope. But what can we do as an organization without putting pressure on the researchers? They already understand the urgency. But what can we do as an organization to accelerate the pace of discovery? And so that's where we as an organization are committed into the next few years to accelerate that piece of discovery in terms of therapies. We're creating new programs that will allow researchers to take what they see in the laboratory under a microscope and potentially bring it into clinical trials. We've recently joined with the Center for Drug Research Development, which is a great center up in Vancouver, who have industry experience. So a lot of the researchers may not really know how to take something they see in a mouse or in cells and potentially bring it to the human body. And so with the Center for Drug Research Development, we hope that we can provide the tools for the researchers to take that therapy into people living with multiple sclerosis. At the same time, we are extremely focused on progressive MS. As I said before, there are currently no therapies, and we as an organization are committed to ensure that we can find therapies in the next 5 years. We're pushing on government to ensure that they put more resources into progressive MS, and as well at the international level, there is the Progressive MS Alliance, where we've joined in with the UK MS Society, the Italian MS Society, and the US MS Society, and other MS societies in terms of finding potential answers for multiple sclerosis in terms of progressive MS. You'll hear more about that in the next few months. There's exciting pieces that are coming through with that. And more importantly, I really hope to come back in the next few years and to provide you more details about the pieces of research that we have funded and where they have gone, but more importantly, that we can come here next year and potentially say that we now have treatments for progressive MS. So I thank you all again and wish you all a very good World MS Day. And I know together as an organization, we truly hope to find an end to multiple sclerosis. So thank you.
Thank you, Dr. Lee. Folks, we're running about 10-15 minutes behind schedule. I hope that you'll bear with us. I'd like to open the floor for questions. I'm going to give the microphone. And if you have any questions, please raise your hand.
This is probably too simple of a question, but I'd like to ask some more about your presentation, Dr. Stetefeld, in terms of what is the importance and the value of changing. I think what you were saying was that you're changing the direction, or you're trying to change the path. So what's the importance or the value, and then the connect to what, then the connect, I guess, to stem cell research and development?
I'm afraid you asked me what's on the board in this Winnipeg Free Press picture. Okay, you know, you can't treat a patient with a natural molecule. It means we take the big guy, which is altogether about almost $500,000. It's a monstrous protein. And we try to we always make a so-called divide and conquer strategy. We cut out the pieces which do the job. And these pieces we develop, or we try to develop drugs, which technically mimic then the job of the monster guy. An issue what we in the moment haven't targeted, we have an interest in compounds which just pass the blood-brain barrier. But we are in the moment not, it's too early to think about really a targeted approach, what Donna was mentioning. But technically this is the way it goes. We have a big system, we try to understand, and we nail it down to a couple of amino acids. And what you have seen is a bit of sensation, which that you can mimic the function of a full molecule with about 7 amino acid residues. And it's not the whole story, but it's a very, very strong starting point. And most probably the reason why the MS Society is funding this research. Was this the question? The answer?
It sounded good to me, but so then with that ability, it makes it easier to then progress with the stem cell research, right? Okay, thanks.
Do we have other questions?
Very interesting presentation. I'm just curious, especially for the stem cell therapy, I know the minimal cost right now run at least at around $50,000, if not higher, for a patient. So I'm just wondering from an application perspective, how feasible, even if it does work, is there any discussion with the government to be able to support, to make sure that the patients do get in the end the treatment if it's available? And just I know from the antibody story, they're very expensive as well, and in many ways they're a huge burden on the system overall. So any thoughts on how to approach that?
Yeah, I've got a couple of answers. The first is that this stem cell therapy is not the same as the other stem cell therapy, which involves my kind of stem cells, young blood-making stem cells, and an approach of giving very high-dose chemotherapy, a long hospital stay, and then recovering up with your own blood-making stem cells. And basically, you think of it a reboot of the immune system. So that's one kind of stem cell. What I talked to you about tonight was mesenchymal stem cells, and that is to basically give a novel way of giving a really strong quieting down of that immune system, and possibly some factors that are going to be really have a positive effect on the healing part of in the brain, where the different neurologic cells are talking to each other. So that's where we think that's going to work. And then there are, as you read through, a ton of other stem cell therapies, and different embryonal stem cells. Like, you guys don't want to stay a couple more hours, so we can just keep going. Never give a professor a microphone. One of the themes that we have, being from Manitoba, is practical cell manufacturing, practical cell therapy. And so we're working very closely with other cell manufacturing programs in Canada, working with Health Canada to try and get an understanding that we can do this in a smart way without really heavy regulatory burden, and let's basically get the job done rather than getting lost in paperwork. So, and the other part is, you know, if we have a good treatment, we're going to figure out how to get it paid for.
Are there any other questions?
Could I ask you, Dr. Wall, about harvesting bone marrow from patients? You said there are a couple of different kinds of stem cells, so from a match or from the patient himself. And how is it harvested? Does it have to be through an aspiration of the bone? Which that hurts.
Yeah, that hurts. So one of the nice things, so if I'm going to answer it first from the type of cells that we're working with, mesenchymal cells, one of the nice things is these guys grow like crazy in the laboratory. Okay, so we can grow them up, grow them up, grow them up. So I can work with a couple of teaspoons of bone marrow. And so what that is like, I'm not going to tell you a story. It's more than a blood test. We give you local numbing to cover the site, and there is an ouch involved because we make some suction on the inside of the bone, and there's some receptors that can feel it. We can't numb that up completely, but it's a procedure that's done in moments. And it's very different than what you may have heard about in the past of a bone marrow harvest for the hematopoietic transplant. There we need to collect a liter of bone marrow. So we're completely, this is very, very different. What you mentioned was transplant from yourself, and that is in the work from the Ottawa team, where they would harvest from the patient themselves, freeze down those cells, and then give that patient really strong chemotherapy to wipe out all the mixed up immune cells that are there, and then give back their own blood cells. They'll recover, and then the hope is that as they recover up and grow back up into all the other parts of the blood-making system, they're not going to make the same mistake twice. They're going to get themselves back on the straight and narrow. Okay, so that's called autologous hematopoietic stem cell transplant. And so that's what I kind of led with when I was talking. There's stem, and then there's a bunch of other different kinds of stems.
This question to Dr. Stetefeld. The netrin, is that any part of the interferons that we are now receiving?
Sorry?
The netrin, we are currently taking interferons. Is that any part of the interferon, the netrin?
No, the netrin is a molecule which is secreted into the extracellular space, and its job in your and in my brain and in the CNS or even in other tissues is to guide cells, to tell a cell to go from A to B. And interferon is, you are talking about the cytokine, the inflammatory response. Netrin is a guidance cue which has nothing to do with this kind of event. But if you would, or let's say the people in pharma industry, they look for a target under with three questions. The first is to kill the pain, and this is most probably the kind of drug prescribed these days for multiple sclerosis pain. You want to diminish the pain for the patients. The highest priority always has, of course, to go for the reason why is the damage. But even, today, Canada is one of the sadly leading countries with cystic fibrosis. No one knows why these patients have this. We find this third kind of effects. And we decided, it's like after this winter that the roads in Winnipeg have a problem. I was very happy that I live close to the universities. My car was broke when I was coming here. Okay, we can't change too much the climate. or the reason why the roads are in bad shape. And this would be the first defect fight, you know. The second defect fight, this is what I'm interested in, to repair or to reinstall rheoline sheath repair. You know, the sheath is damaged out of a reason which we in the moment don't understand, but we try to bring the workers, the machineries in place who produce rheoline and take care that it's properly inserted, so to speak. And the third defect is always, you know, you would bring the car to a mechanic and he repairs it. But natrine as such has nothing to do with the cytokine or interferon signaling as an inflammatory response.
For Dr. Wall again, sorry about that, wait a minute. Just a quick question. When you're doing your stem cell research, you're doing it from bone marrow, whatever, with your mesenchymal cells. What's the difference between using, let's say, stem cells from adipose tissue? I mean, is there anything?
Probably not a lot.
Okay.
So you can get these mesenchymal cells from tooth lining, from hair follicles, from skin, from fat. Liposuction is a tremendous source of mesenchymal cells. I'm a blood doctor, so we get it from the bone marrow. So.
Thank you for posing questions to our esteemed panel of doctors. We really appreciate their knowledge and expertise. As our research reception draws to a close, I'd like to thank our esteemed speakers for providing us with new and helpful information about MS. MS is a mysterious disease. While we still don't know what causes MS, the last decade has seen encouraging advances in disease and symptom management, enhancement of function, and improvement in the quality of life. A great deal has been accomplished by our dedicated researchers in Manitoba and across Canada, and their advances continue to offer hope for everyone affected by MS. Thank you for supporting the 2nd Manitoba Division MS Research Reception.